Rabies is one of the deadliest and most interesting diseases known to man. While it does not have the same worldwide impact as smallpox, polio, or the plague, it has remained an elusive and sinister disease. It is an ancient disease having been documented since 2000 BC. It was a source of terror for people due to its devastating symptoms. Dogs were depicted as the primary sources of the disease and were identified as capable of transferring this scourge to humans. For example, in the Mesopotamian Codex of Eshnunna in 1930 BC, the owners of dogs showing characteristic symptoms of rabies were advised to take preventative measures against bites. Although we have a better understanding of rabies today, the fear associated with the disease remains intact because although preventative measures have changed, the distressing and fatal consequences of the disease have remained the same. So what is rabies? Rabies is a disease of the central nervous system caused by the Lyssavirus genus of viruses, which is part of the rhabdovirus family. It is a bullet-shaped virus containing single-stranded negative sense RNA. The RNA is associated with the ribonucleoprotein complex and is enclosed within an outer envelope. The genome of the virus contained within the RNA is composed of only 5 genes that encode 5 proteins. The 5 encoded proteins are nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and the viral RNA polymerase. The order of each gene in relation to the others is highly conserved and is what determines the structure and function of the rabies virus. According to the World Health Organization, it is estimated that rabies causes 59,000 human deaths annually, with 95% of cases occurring in Africa and Asia. In these countries, dogs are responsible for 99% of rabies cases, and most cases are present in rural areas and in children under 15 years of age. Importantly, dog-mediated rabies has been eliminated from many places, including Canada. Since 1924, Canada has only had 25 confirmed cases of rabies, although rabid animals are still a concern, with the majority being raccoons. The rabies virus is most commonly found in terrestrial mammals such as dogs, bats, monkeys, and raccoons. It is transmitted to humans via bites from infected animals. It is present in the saliva of the animal and is hence transferred to the new host through the bite. However, there have been rare cases where the virus has been transmitted via sexual intercourse or from mother to child through breastfeeding. After transmission, the rabies virus travels from the site of the bite to the brain by moving through the nerves of the new host. It travels from the peripheral nervous system to the central nervous system, finding its way to the brain where it causes symptoms to begin to develop, ultimately culminating in fatal encephalitis. Once characteristic symptoms of rabies are evident in a patient, death is almost always imminent. Early signs of rabies can be similar to those of the common cold. As the disease advances, other symptoms like fever, nausea, agitation, confusion, difficulty swallowing, and itching at the site of the bite are usually seen. Upon further progression, abnormal behavior is observed, such as the most characteristic symptom of rabies and perhaps the most infamous, hydrophobia or fear of water. This is brought on by difficulty in swallowing fluids. Hydrophobia is the main symptom associated with encephalitic rabies. In order to identify host cells and carry out replication within the cell, the rabies virus has approximately 400 spikes on its surface. The virus aggregates as an endosome within the cell. Viral membranes then fuse to the endosomal membranes, and viral contents are expelled into the cytoplasm. Since the virus genome consists of a single-stranded negative sense RNA, mRNA must first be synthesized. The L gene of the virus encodes the viral polymerase, which leads to the transcription of rabies RNA into mRNA within the nucleus of the host cell. The mRNA is then translated into the N, P, M, G, and L proteins on ribosomes in the host cytoplasm. The viral units exit the cell to infect other host cells by moving along until the virus reaches the central nervous system, ultimately causing inflammation and eventual death of the afflicted individual. Unfortunately, there is no available test to detect rabies virus in humans before the onset of clinical disease, and the diagnosis is made when the patients display classical symptoms such as hydrophobia, and by the time symptoms are shown, nothing can be done to stop the disease. Most cases of rabies are confirmed during the clinical disease stage or post-mortem. Post-mortem tests that can be used include the direct fluorescent antibody test, electron microscopy, and nucleic acid tests. Rabies can be confirmed by detecting viral antigen using direct fluorescent antibody testing. In this test, when rabies is present in tissue, the rabies antigens will bind to antibodies and produce fluorescent signals under a microscope. Electron microscopy can also be used to detect the presence of rabies using the whole virus. The virus forms round or oval inclusions called Negri bodies within the cytoplasm of the infected nerve cells. These Negri bodies can be visualized under an electron microscope. Lastly, nucleic acids can be used to confirm the results of a DFA test or to detect the virus in infected tissues. Sometimes samples such as saliva or skin biopsies contain only small amounts of rabies, which makes it difficult to detect. By using reverse transcriptase, we are able to copy the rabies viral RNA into DNA, which can then be amplified using polymerase chain reaction. So far, the only effective treatment for rabies is post-exposure prophylaxis. This includes extensive washing and local treatment of the bite immediately after exposure, as well as immediate vaccination and administration of rabies immunoglobulin to induce the production of rabies-neutralizing antibodies against the virus. You may have heard horror stories about rabies vaccines being 20 or so painful needles to the abdomen. This actually was the case back in the 1980s with the nerve tissue rabies vaccine, but worry not, today's vaccine is a 4 to 5 shot regimen given intramuscularly. If you have been exposed to the rabies virus, you would also receive rabies immunoglobulin injections. I actually had to get the rabies shot personally a few years ago after I was bitten by a cat, and while it definitely wasn't the best experience of my life, luckily it is nothing like the horrors I imagined and had heard about. The nerve tissue rabies vaccine is not recommended for use by the World Health Organization anymore, and very few countries still use it. Besides being a horrible experience, it simply wasn't as effective as the current vaccine, and it had many unwanted side effects. Post-exposure prophylaxis can effectively prevent the onset of symptoms and the virus. However, once the virus enters the central nervous system, it will eventually cause death of the patient. In general, prognosis is extremely poor and death occurs within a few days. Although we don't currently have any medications to treat rabies, perhaps inducing a coma is the answer. A 15-year-old girl who contracted rabies from a bat was put into an induced coma, which allowed her native immune response to the virus to mature. 5 months later, she survived. However, she was left with some neurological deficits such as dysarthria and unsteady gait. Regardless, this information might help us combat rabies in the future and is still a unique and favorable result.